water and act as a source of infection. Thus, the atmospheric water and the surface water get usually contaminated from these different sources, and the groundwater are relatively less frequently contaminated from these contaminants. Now we will discuss about the significance of microorganisms in water. What is the significant role of microorganisms in water? We will discuss. The microorganisms in water play dual role, that is significant as well as harmful, or beneficial as well as harmful. Pathogenic pathogens in water causes various diseases to human beings, plants, and animals. Some microorganisms may produce toxic metabolic products in water. Many microorganisms in water catalyzes ecologically significant biochemical reactions. Some of these are described as decomposition of organic matter, photosynthesis, nitrogen fixation, biochemical activities of chemo, autotrophs, and denitrification. The decomposition of organic matter, the microorganism degrade organic matter in the water and sewage to produce stable inorganic compounds or products. These reduces BOD of water bodies in which it is added, that unless create oxygen deficiency, a harmful condition for aquatic life. The minerals produced in this process may be used by plants and autotrophic microorganisms. The oil spills in the water that are harmful to aquatic organisms are digested by lipolytic bacteria and fungi. Photosynthesis. The algae and photosynthetic bacteria are primary producers in aquatic habitat. They synthesize carbohydrates using minerals, atmospheric carbon dioxide as carbon source, and sunlight as energy. The oxygenic photosynthesis in water by cyanobacteria helps to maintain oxygen level in the aquatic habitats. Nitrogen fixation is the third point. The nitrogen fixing algae and bacteria in aquatic habitat utilizes atmospheric nitrogen for their growth and also provides a fixed nitrogen to the other growing organisms. The photosynthetic diazotrophs are nutritionally very simple, which grows at expense of mineral water with sufficient availability of sunlight. Then we'll discuss about the next point, that is different indicator microorganisms or index microorganisms, index of water pollution, index organism or indicator organisms in the water pollution. Coliforms are considered as index microorganisms. For that, we have to analyze water to show the load of microorganisms. A major quantity of earth water is salt. Earth's water is salty, and major quantity of remaining water is not potable due to presence of toxic chemical and microorganisms. It is estimated that about 0.007% of the water is available for direct human consumption. The drinking water is obtained from rivers, streams, lakes, wells, and bore wells. Such natural water supply, particularly the surface waters, are polluted with domestic and industrial waste. The water is a potential carrier of pathogenic microorganisms, which may hazard the health of living beings. According to rough estimate, more than 15 million deaths worldwide resulted annually for waterborne infection. Diarrheal diseases of small children alone result in more than 2 million deaths annually in developing countries. The outbreak of cholera or other diseases may also cause deaths. Hence, the drinking water must be free from microorganisms, particularly the pathogens. The number of saprophytic bacteria also should not exceed 100 ml of water. These diseases, many diseases are transmitted through drinking water, especially the infections of intestinal tract. Some of the common examples of enteric diseases are typhoid, dysentery, amoebic dysentery, cholera. The causative organism of these diseases are related through released through fecal matters of the patient and may enter into drinking water source. Hence, microbial analysis of water is essential and index of water pollution or different indicator organisms should be studied. So, the coliforms are are the group of intestinal or enteric bacteria characterized by lactose fermentation at 37 degrees Celsius within 24 hours with production of organic acid and gases, carbon dioxide and hydrogen by mixed acid fermentation pathway. They produce pink color colonies on McConkey's agar, whereas the lactose non-fermenting organisms produce colorless colonies. They are gram-negative short rods. Escherichia coli or coliforms are gram-negative short rods and show pink color colonies on McConkey's agar plate containing lactose as a source of carbon. The pathogen causing waterborne diseases enter into water bodies via intestinal discharges of human and other animals suffering from the disease. Certain bacterial species, particularly Escherichia coli and other coliforms, that is fecal streptococci, Streptococcus faecalis, and Clostridium perfringens, are normal inhabitants of large intestine of human and other animals and are frequently released in feces. It is estimated that each person discharges about 100 to 100 to 400 billions of coliform bacteria per day. The fecal streptococci can be detected in water samples using selective media such as sodium azide glucose broth. Sodium azide inhibits gram-negative bacteria. Presence of these bacteria in water can be confirmed by streaking a loopful of positive sodium azide glucose broth tube on ethyl violet azide glucose agar. Ethyl violet and sodium azide inhibits virtually all bacteria except fecal streptococci. The index organism, that is the particular organism that should be detected for safe drinking water, to remark as a safe drinking water, is the index organism. These organisms are specifically considered as coliforms, that are fecal coliforms that discharged by the humans or animals daily into their feces, and they contain Escherichia coli, Streptococcus faecalis, and Clostridium perfringens at higher amounts. So, if we detect Escherichia coli that is having lactose fermentation capacity and pink coloration in McConkey's agar, Streptococcus faecalis that is having growth only on ethyl violet and sodium azide glucose agar specifically. So, and the Clostridium species, then we can detect that the water supply is being contaminated with the fecal source or sewage. So we can avoid that and can purify that water source. The species of Clostridium or Clostridium perfringens being anaerobic requires specific technique for detection and isolation. Bifidobacteria may enter in water from other sources than fecal matter. Enterobacter erosion, although present in human and animal intestine, it is not found on grains, and it may also found on grains and plants. Presence of these bacteria in drinking water bodies do not confirm the fecal pollution. However, Escherichia coli being strictly intestinal, their presence in the water assures fecal pollution. E. coli can be easily isolated from water using selective medium like McConkey's agar. Hence, the E. coli is the most perfect index organism or indicator organism in the water pollution. These bacteria are differential from Enterobacter erosion on the basis of Emwick test and Ejackman test. The presence of coliform, especially Escherichia coli in the water indicates fecal pollution. If these organisms are present in water, there is high probability of presence of intestinal pathogens. Water analysis techniques have been developed for the demonstration of these bacterial species. For a healthy person do not excrete pathogenic organism, but they appear in water through the fecal matters of the patient suffering from the intestinal infections. So the index of water pollution is dependent upon the indicator organism, that is Escherichia coli, Streptococcus faecalis, and Clostridium perfringens. But the coliform bacteria, specifically Escherichia coli, is the index organism for detection of water pollution, as it is easy to identify and it is strictly related with the intestine of the human beings. And the patients suffering from the intestinal infections release these bacteria more frequently than others. So in the next point, we will discuss about the microbial examination of water.